Chapter 14, Mendel and the Gene Idea. In the last chapters, we covered the life cycles of the cell and the roles of mitosis and meiosis. In discovering these life cycles, we were also introduced to chromosomes and their role in heredity. In this chapter, we will dive deeper into those units of heredity called genes. In this lecture, students will model the processes and patterns of inheritance in eukaryotic organisms. Particularly, you will describe and model the fundamentals of genes and alleles. Describe the inheritance using proper genetics terminology pertaining to organization and transmission. Predict the process of inheritance from numerical and/or pedigree data. Produce numerical predictions and/or pedigree patterns for the processes of inheritance. And describe and relate the contribution of environmental variation to phenotypic variation. To begin any discussion on genes and heredity, we have to talk about the father of modern genetics, Gregor Mendel. Mendel was an Austrian monk and scientist whose careful work in the mid to late 1800s breeding pea plants established many of the rules of heredity now referred to as the laws of Mendelian inheritance. The particulate hypothesis is the idea that parents pass on discrete heritable units. Today we call these heritable units genes, but when Mendel was conducting his experiments, he did not know what these heritable units actually were. There were a number of distinct advantages of using pea plants for genetic study. There are many varieties with distinct heritable features or characters, such as flower color. We call these character variants, such as purple or white flowers, traits. Mating can be easily controlled. Each flower has sperm-producing organs called stamens and an egg-producing organ called the carpel. And cross-pollination, which is fertilization between two different plants, involves dusting one plant with pollen from another. There are many different characters associated with pea plants, but Mendel chose to track only those characters that occurred in two distinct alternative forms. He also used varieties that were true breeding, which are plants that produce offspring of the same variety when you continue to self-pollinate them. This means if you breed a purple flower with itself, you will always get purple flower plants as offspring. In a typical experiment, Mendel mated two contrasting true breeding varieties, a process called hybridization. Here we see he used purple flower true breeders and white flower true breeders. The true breeding parents are called the P generation, which stands for parental generation. The hybrid offspring of the P generation are called the F1 generation. When those F1 individuals self-pollinate or cross-pollinate with other F1 hybrids, the F2 generation is produced. Let's look at the following colors produced. When Mendel crossed contrasting true breeding white and purple flowered pea plants, all of the F1 hybrids were purple. But when Mendel crossed those purple F1 hybrids, many of the F2 plants had purple flowers, but we also see the re-emergence of the white flower trait. Through these experiments, Mendel discovered this ratio of 3:1 in the F2 generation. In fact, Mendel observed the same pattern of inheritance, this 3:1 ratio, in 6 other pea plant characters, each represented by 2 traits. Consider this a golden ratio in genetics. We will talk about 2 golden ratios in this lecture. Mendel reasoned that only the purple flower factor was affecting the flower color in the F1 hybrids. Mendel called the purple flower color a dominant trait and the white flower color a recessive trait. The factor for white flowers was not diluted or destroyed because it reappeared in the F2 generation. What Mendel called a heritable factor is now what we call a gene. We know from the last chapter that genes are the units of heredity made up of segments of DNA. However, keep in mind that Gregor Mendel was born in 1822 and conducted his pea plant experiments in the 1850s and 1860s. DNA's role in genetic inheritance wasn't discovered until 1943, nearly a century later. Mendel is called the father of modern genetics because his discoveries were the first in the field of genetics that set us on a course of discovering how inheritance works. So let's look at Mendel's model of inheritance. Mendel developed a hypothesis to explain the 3-to-1 inheritance pattern that he observed in F2 offspring. First law: alternative versions of genes account for variations in inherited characters. These alternative versions of a gene are now called alleles. Take a look at the picture of chromosomes. This is a pair of homologous chromosomes. Remember, homologs are the mom and dad versions of a chromosome. Chromosomes in a homologous pair are the same length and shape and carry genes controlling the same inherited characters. Students often confuse character, trait, gene, and allele. The character we are looking at is flower color. The two versions of the gene for flower color are purple and white. So we have a white allele and a purple allele. Also notice that the two alleles are in the same place on the chromosome, the same locus. Remember, this is very important for when homologs cross over during prophase 1 of meiosis. Here we can see the actual genes, the DNA segments that make up these two different alleles. The allele for the purple flower contains a gene that makes an enzyme to make a purple pigment. This is why flowers carrying this allele have purple petals. In contrast, the allele for the white flower contains a gene that results in no enzyme. Therefore, no purple pigment is made and the flowers are white. Mendel's second law: for each character, an organism inherits two alleles, one from each parent. The two alleles at a particular locus may be identical, as in the true breeding plants in Mendel's pea generation. In this image, the two alleles are symbolized by uppercase P for purple and lowercase p for white. The true breeding purple flower in the pea generation has two uppercase P alleles. The true breeding white flower in the pea generation has two lowercase p alleles. Alternatively, the two alleles at a locus may differ, as in the F1 hybrids. These flowers are purple just like the true breeders in the pea generation. However, the F1 generation purple flowers are hiding something. These flowers have one uppercase P and one lowercase p, so one copy of each allele. Mendel's third law: if the two alleles at a locus differ, then one, the dominant allele, determines the organism's appearance and the other, the recessive allele, has no noticeable effect on appearance. We see this here with the purple flower in the F1 generation. The uppercase P representing the purple allele is dominant over the lowercase p representing the white allele. Mendel's fourth law, now known as the law of segregation: the two alleles for a heritable character separate or segregate during gamete formation and end up in different gametes. Thus, an egg or a sperm gets only one of the two alleles that are present in the organism. We already know this happens because we learned meiosis, which separates homologs, then sister chromatids, to produce haploid cells. This is necessary so that when the sperm and the egg meet during fertilization, the diploid chromosome number is restored. Mendel's segregation model accounts for the 3-to-1 ratio he observed in the F2 generation of all of his crosses. The possible combinations of sperm and egg can be shown using the Punnett square, a diagram for predicting the results of a genetic cross between individuals of known genetic makeup. A capital letter represents a dominant allele, and a lowercase letter represents a recessive allele. Take a look at the Punnett square shown in the bottom right. It's very important for you to understand how to set up and use a Punnett square. They will get larger later in the lecture. This Punnett square is crossing two F1 generation purple flowers, each with the genotype big P, little p. Across the top of the Punnett square are the sperm alleles from the male plant. Due to Mendel's law of segregation, the alleles have been separated into two separate sperm. We see one big P and one little p across the top. On the left side, we see the egg alleles from the female plant, which have also been segregated. Again, we have one big P and one little p. Now we fill in the square. The top left box gets two big P's. The top right box gets one big P, one little p. The bottom left box gets one big P, one little p, and the bottom right box gets two little p's. The big P is dominant over the little p, and therefore we get 3 purple flowers and 1 white flower. The white allele is recessive, and therefore to express the white color trait, this individual needs both alleles to be the recessive white allele. Now we see why Mendel observed the pattern of this golden 3-to-1 ratio in the F2 generation of his crosses. If that was a little bit confusing, we're going to go over it one more time, this time using the correct genetic terms. Here are the 4 F2 generation individuals created after crossing the hybrid F1 generation. On the left, we see the 3-to-1 ratio represented. 3 purple flowers exhibiting the dominant trait and 1 white flower exhibiting the recessive trait. The physical appearance of these flowers is called the phenotype. My hint to remember this is physical and phenotype both begin with the PH. The phenotype is distinguished from an organism's genotype, which is the genetic makeup of the individual. Take a look at the right column showing the genotypes of the 4 individual flowers. We see 2 new terms here, homozygous and heterozygous. An organism with 2 identical alleles for a character is said to be homozygous for the gene controlling that character. Where do you see homozygous genotypes here? You should find 2. One individual is homozygous for the dominant trait, big P, while the bottom individual is homozygous for the recessive trait, little p. An organism that has 2 different alleles for a gene is said to be heterozygous for the gene controlling that character. Where do you see heterozygous genotypes here? There are 2 purple flowers with the genotype big P, little p. Now notice the ratio for the genotypes of the F2 generation, 1 to 2 to 1. Please note this very important ratio as well. This means there is 1 homozygous dominant individual, 2 heterozygous individuals, and 1 homozygous recessive individual. Because of the different effects of dominant and recessive alleles, an organism's traits do not always reveal its genetic composition. One cannot tell the genetic makeup of a purple flower just by looking at its phenotype. Unlike homozygotes, heterozygotes are not true breeders. To bring it back to the original image, what do you notice about the genotype of the individuals in the P generation? Both are homozygous. We have one that is homozygous dominant and the other is homozygous recessive. Remember, we called these P generation individuals true breeders. All true breeders are homozygous. Crossing the two different true breeding flowers created a hybrid flower in the F1 generation. It is a hybrid because it is heterozygous. It contains one copy of each allele. Therefore, heterozygotes are not true breeding. The recessive allele is hiding behind the dominant purple phenotype. But when we cross two of these F1 generation individuals, the recessive white allele re-emerges. The F2 generation here has a phenotypic ratio of 3:1 and a genotypic ratio of 1:2:1. Now we see how an individual with the dominant phenotype, like the color purple here, could be either homozygous dominant or heterozygous for the genotype. So how would we figure out whether a purple flower was homozygous dominant or heterozygous? The answer is to carry out something called a test cross. In a test cross, we breed the mystery individual with a homozygous recessive individual. In this example, we have a purple flower with an unknown genotype, and we are test crossing it with a white homozygous recessive flower. Then all you have to do is look at the color of the offspring. If any offspring display the recessive phenotype of white flowers, the mystery purple flower parent must be heterozygous. Let's take a look to see how. We have two possible outcomes here. Either the purple parent is homozygous dominant, big P, or it is heterozygous, big P, little p. The Punnett square on the left shows if the parent is homozygous dominant. The alleles for the sperm, in this case the white homozygous recessive flower, have been segregated across the top, and the alleles from the egg, in this case the purple flower with the big P, have been segregated on the left side of the square. After we fill in the square, we see that all offspring are purple. There are no white individuals. What are the genotypes present in these offspring? All offspring here are heterozygous hybrid individuals. Now look to the Punnett square on the right. This square shows the outcome if the purple parent flower is heterozygous, big P, little p. Again, the sperm for the white homozygous flower is segregated across the top, and the alleles from the egg are on the left, big P, little p. After we fill in the Punnett square, we see there are two purple flowers and two white flowers, a 50-50 or 1:1 ratio here. The white flower color has re-emerged, no longer hiding behind a dominant allele. This test cross has revealed that the purple flower was indeed heterozygous. Recall that Mendel's fourth point in his hypothesis, now known as the law of segregation, states that the two alleles for a heritable character separate or segregate during gamete formation and end up in different gametes. Thus, an egg or a sperm gets only one of the two alleles that are present in the organism. We saw this while creating our Punnett squares. Mendel derived the law of segregation by following a single character. The F1 offspring produced in the cross were monohybrids, which are individuals that are heterozygous for one character. The word mono, of course, means one. And they are hybrids because they contain both the dominant and recessive allele. A cross between these heterozygotes is called a monohybrid cross. This purple and white flower example is a monohybrid cross. Mendel identified his second law of inheritance by following two characters at the same time. We see this Punnett square has 16 squares instead of 4. Let's look closer at this example. We begin with two different true breeders. The dominant true breeder is yellow and smooth. It is homozygous dominant for color. Yellow here is dominant, denoted by the big Y, and homozygous dominant for texture. Smooth is dominant here, denoted by big R. The recessive true breeder is green and wrinkled. It is homozygous recessive for color. Green here is recessive, denoted by the little y, and homozygous recessive for texture. Wrinkled here is denoted by the little r. Crossing these two true breeding parents, differing in two characters, produces dihybrids in the F1 generation, heterozygous for both characters. Here the dihybrid individual is big Y, big R. A dihybrid cross, a cross between F1 dihybrids, can determine whether two characters are transmitted to offspring as a package or independently. Using a dihybrid cross like this, Mendel developed the law of independent assortment. You learned about independent assortment in Chapter 13 while discussing meiosis. The law of independent assortment states that each pair of alleles segregates independently of each other pair of alleles during gamete formation. Strictly speaking, this law applies only to genes on different non-homologous chromosomes or those far apart on the same chromosome. Genes located near each other on the same chromosome tend to be inherited together. This is called linkage, and we'll learn more about linked genes in the next chapter. Let's return to this dihybrid cross for a moment. Again, this is called a dihybrid cross because we are looking at two characters, color and texture. The individuals in the P generation have two alleles for each character, so we see 4 alleles here. I'm going to walk you through how to complete a dihybrid cross. Step 1 is to identify the parents. Let's look at the homozygous dominant individual, big Y, big R. This individual exhibits the dominant phenotypes for both traits, yellow and smooth. The other individual is the homozygous recessive, little y, little r. This individual exhibits the recessive phenotypes for both traits, green and wrinkled. Step 2 is to create all gametes possible for each parent. These are both true breeders. True breeders only produce one kind of gamete. We see the homozygous dominant individual creates the gamete big Y, big R, and the homozygous recessive individual creates the gamete little y, little r. Step 3 is to set up the Punnett square. The parental cross does not need a Punnett square because there's only one combination possible as an offspring. When we fertilize the sperm and egg gametes from the P generation, we create the dihybrid big Y, big R. This is our F1 generation. Great, now we have to repeat the steps again for the next cross, the dihybrid cross. In the dihybrid cross, we cross 2 of the F1 individuals. We've already accomplished step 1. Step 2, create all possible gametes. Creating the gametes for the dihybrids is more complicated than it was for the P generation. To figure out the F1 gametes, we need to use a method called FOIL, which means first, outer, inner, last, FOIL. You may remember this from math class. In this example, we have a dihybrid using the letters A and B. Start with the first letter, capital A. Always remember when we make gametes, we want one copy of each character. That way, that individual has genetic material for the characters. Notice that every gamete has one copy of an A and one copy of a B. The FOIL method helps us to ensure that we create every possible type of gamete from the genetic material of the parent. So start with capital A and draw arrows across the top to each B allele. Those are your first 2 gametes. You have big A, big B, big A, little b. Next, start at the lowercase a and draw arrows along the bottom to each B allele. Those are your last 2 gametes, little a, big B, and little a, little b. This individual was able to create 4 distinct gametes. Back to our example, it is very similar to the one that we just used. The F1 generation is able to create 4 gametes: big Y, big R, little y, big R, and little y, little r. Because we are crossing the F1 generation by itself, both the sperm and the egg make these 4 gametes. So step 3, set up the Punnett square. The 4 gametes for the sperm are across the top, and the 4 gametes for the egg are across the side. Step 4, fill in the Punnett square. This is the fertilization process. So chromosome number and therefore allele number is restored here. Ensure that you have 2 copies of alleles for each character. Looking at the first box in the Punnett square, the gamete from the sperm is big Y, big R, and the gamete from the egg is also big Y, big R. This individual is therefore big Y, big Y, big R, homozygous dominant for both traits. A quick word on this: always write the capital dominant allele before any lowercase recessives. Also, always keep the order of the letters the same. In this case, Y and then R. And of course, keep the alleles for the same traits together. You wouldn't write this as big Y, big R, big Y, big R. And finally, step 5, analyze. Look for the ratios. This is a dihybrid cross, so we have a new golden ratio here, 9:3 to 3:1. So what's 9:3 to 3:1? Pause and try to find this. Is this a genotype or a phenotype ratio? 9:3 to 3:1 is the phenotype ratio, the physical appearance. There are 9 individuals who are dominant for both characters, yellow and smooth, 3 individuals who are dominant for color but recessive for texture, so yellow and wrinkled, 3 individuals who are the reverse, so recessive for color but dominant for texture, that's green and smooth, and 1 individual who is recessive for both characters, green and wrinkled. You need to know the 9:3 to 3:1 phenotypic ratio for dihybrid crosses. However, you do not need to know the genotype ratio for dihybrid crosses. Here is a table of monohybrid and dihybrid crosses with their respective phenotype and genotype ratios. Write this down and know it well. You'll certainly see questions about it on exams. Now that we've properly walked through the dihybrid cross, we can return to the concept of independent assortment. Remember, Mendel identified the law of independent assortment by following 2 characters at the same time, just as we've just done in the dihybrid cross. This image shows 2 possibilities. The left shows a Punnett square if the alleles did not exhibit independent assortment and therefore were not able to randomly rearrange to form various gametes. On the right, we see a Punnett square where the alleles have gone through independent assortment. What you need to notice to grasp this concept is the parental gametes versus the F1 gametes. On the left, if no independent assortment was allowed to occur, then alleles that were inherited together in a gamete would remain together as they got passed down the generations. The parental gametes are big Y, big R, and little y, little r. So they would be inherited as packets like this by the F1 generation, and then when the F1s were crossed to create F2, the gametes would still be packaged together. Look at the Punnett square on the left. Notice the gametes, big Y, big R, and little y, little r, the very same as the parental generation gametes. No independent assortment has occurred. Mendel hypothesized that if this is how inheritance works, then the phenotypes of the F2 generation would exhibit a 3:1 ratio, just like the monohybrid crosses yielded. However, this is not what Mendel's dihybrid crosses yielded for offspring, the true phenotype ratio was a 9:3 to 3:1, indicating that the alleles were able to assort independently and rearrange into distinct new variations of gametes. This is showcased in the Punnett square on the right. This is how Gregor Mendel derived the law of independent assortment. Take a moment to review monohybrid crosses and dihybrid crosses. Make sure you understand what is meant by P, F1 and F2 generations, homozygous and heterozygous, dominant and recessive, how to create gametes, the important ratios in genotype and phenotype, and the law of independent assortment. Now you're well acquainted with the concept of dominant and recessive traits. Here are a few dominant traits and diseases in humans. Achondroplasia, the scientific name for dwarfism, is actually a dominant trait. People who do not have achondroplasia are therefore homozygous recessive for the trait. Polydactyly, the scientific term for extra fingers and toes, is also a dominant trait, at least in some cases. Therefore, people with 5 fingers and 5 toes on each hand and foot are homozygous recessive as well. And finally, Huntington's disease is a neurodegenerative disease. You can see from these examples that just because an allele is dominant does not mean that it will be found more frequently in nature. Here are a few recessive traits and diseases in humans. Cystic fibrosis, a potentially fatal disease in which carriers have a defective chloride channel resulting in higher intracellular chloride concentrations, results in an extracellular environment containing more mucus, which makes the cell more viscous, especially in the lungs. Sickle cell disease is caused by an abnormal hemoglobin protein resulting in a sickled cell, a red blood cell, which cannot properly transport oxygen. Homozygous recessive is the full-blown form of this disease, while heterozygotes have a sickle cell trait in which about 50% of the red blood cells are sickle cells and 50% are normal. Finally, there is Tay-Sachs disease, a fatal disease in which a dysfunctional enzyme causes an accumulation of lipids in the brain. Okay, time for a little bit of math. Probability is the branch of mathematics that describes how likely something is to occur. The probability of an event is a number between 0 and 1, where roughly speaking, 0 indicates impossibility of the event and 1 indicates certainty of the event. The higher the probability of an event, the more likely it is that that event will occur. The laws of probability also govern Mendel's law of segregation and independent assortment. When tossing a coin, the outcome of one toss has no impact on the outcome of your next toss. In the same way, the alleles of one gene segregate into gametes independently of another gene's alleles. There are 2 rules of probability that we use to calculate likelihood of inheritance: the multiplication rule and the addition rule. The multiplication rule states that the probability that 2 or more independent events will occur together is the product of their individual probabilities. Probability in an F1 monohybrid cross can be determined using the multiplication rule. Segregation of alleles in a heterozygous plant is like flipping a coin. Each gamete has a 50% chance of receiving the dominant and a 50% chance of receiving the recessive allele. Use this representation of a monohybrid cross as a visual for this. Remember, a monohybrid cross is a cross between 2 hybrid individuals, containing 1 dominant and 1 recessive allele for just 1 character. Here the alleles are big R and little r. When the individuals undergo meiosis to create haploid gamete cells, the alleles which are present on homologous chromosomes separate or segregate in the process. For both hybrids with the genotype big R little r, there are therefore 2 possible gametes, one containing the big R and the other containing the little r. This is where the coin toss comparison comes into play. Just like you have a 50% chance of getting heads or a 50% chance of getting tails, the likelihood of getting the big R gamete or the little r gamete is 1/2 or 50% each. Both of our parental individuals are hybrids, therefore this is true for both of them. When we set up the Punnett square, the gametes are placed along the top and left sides. Now this is where the math comes in. According to the multiplication rule, the probability that two or more independent events will occur together is the product of their individual probabilities. To rephrase that, for this example, the probability that the big R from the sperm and the big R from the egg will occur together in an individual is the product of their individual probabilities. So, to figure out the likelihood of an individual having the genotype big R, we multiply the chance of big R, which is 1/2, times the chance of big R, which is 1/2. 1/2 times 1/2 is 1/4. We do this for all other squares within the Punnett square. The likelihood of an individual having the genotype big R little r is 1/2 for big R and 1/2 for little r, which also equals 1/4. Same for the other square containing little r and big R. Finally, the likelihood of an individual having the genotype little r is 1/2 times 1/2, which is also 1/4. And just to tie in points from earlier in the lecture, what are the phenotype and genotype ratios here? Big R is the dominant, and there are 3 squares that would have a dominant phenotype: big R, big R, big R, and little r, big R. To calculate this, 1/4 plus 1/4 plus 1/4 is 3/4. Little r is the recessive phenotype, which accounts for 1/4 of the individuals. This is our 3-to-1 ratio for phenotype. And what about our genotype ratios? 1/4 are big R, 1/2 are big R, and 1/4 are little r. This is our 1-to-2-to-1 ratio. That wasn't so bad, right? Well, it does get a little more complex as we add more characters. We just used a monohybrid cross in our example. However, what if we wanted to explore the probabilities of 2, 3, or even 12 different traits occurring together? A monohybrid cross is small enough that in truth you don't even need to use the multiplication rule. You could just as easily draw a Punnett square. But we saw how the Punnett square quadrupled in size with the dihybrid cross, looking at just 2 characters. Can you guess how many squares a Punnett square looking at 3 characters would require? Did you guess 64? This is called a trihybrid cross, by the way, and it's a lot of work. This is why we use math. So let's try our hand at a few math problems using the multiplication rule. We can apply the rules of probability to predict the outcome of crosses involving multiple characters. Again, characters are things like eye color and hair color. A cross with more than one character is just like 2 or more independent crosses occurring simultaneously. To calculate the chances for various genotypes, each character is considered separately, and then the individual probabilities are multiplied. Let's walk through this example. Here we are given 2 parents. Notice they each carry 2 alleles for 3 different characters denoted by P, Y, and R. We are going to see how to calculate the probability that these parents will produce an offspring that is little p, little y, big R, little r. Okay, here we are again with our 2 parents. I color-coded the 3 different characters to help you follow along. Again, we want to calculate the probability of these parents producing an offspring with a genotype little p, little y, big R, little r. There are 4 steps to follow. Step 1, identify the parental genotypes. We've already done this. Step 2 is to make mini Punnett squares for each character. For this example, we need to make 3. 1 for P, 1 for Y, and 1 for R. They are shown color-coded on the right. Each parent went through meiosis, where alleles segregated to make the individual gametes. Step 3, calculate genotype frequencies. This step is just like what we did with the monohybrid cross Punnett square. Step 4, plug in the genotype frequencies and multiply. We are calculating the likelihood of these parents creating an offspring with a genotype p, little y, big R. Looking at our Punnett squares and genotype frequencies, we see that the probability of p is 1/4. The probability of little y is 1/2, and the probability of big R is also 1/2. When we multiply these numbers, we get a probability of 1/16. Try to use this example to help calculate the probabilities for the remaining offspring variations shown on this slide. That was the multiplication rule. There's another probability rule called the addition rule. The addition rule states that the probability that any one of two or more mutually exclusive events will occur is calculated by adding together their individual probabilities. Mutually exclusive means two events that cannot happen at the same time. For example, the rule of addition can be used to figure out the probability that an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. Returning to our coin flip monohybrid Punnett square, to figure out the likelihood of a plant being heterozygous rather than homozygous, we will use the addition rule. You've probably already figured this out in your head without even realizing you were using the law of probability to do it. To calculate the probability of an individual being heterozygous, you added the 1/4 probability from the big R individual with the 1/4 probability from the little r big R individual. Now remember, the definition of the addition rule is the probability of two or more mutually exclusive events occurring, which is calculated by adding together the individual probabilities. How are the two numbers that you just added up mutually exclusive? They're both heterozygous events, right? They are, however, they're still not the same. The big R individual in the top right has received a big R from the egg and a little r from the sperm, while the little r big R individual in the bottom left has received a little r from the egg and a big R from the sperm. These are in fact not the same. These two variations of heterozygotes cannot occur at the same time, therefore they are mutually exclusive. However, they are both still considered heterozygotes, and therefore we add them together to figure out their probability of occurrence. We can do the same thing to figure out the probability of homozygotes. So we've covered Mendelian genetics. We learned about the heritable units Gregor Mendel discovered, which are now called genes. We covered the particulate hypothesis he developed on how traits are inherited, as well as the law of segregation and the law of independent assortment. We covered monohybrid crosses and dihybrid crosses, and we figured out how to use math to quickly figure out probabilities without needing to draw giant Punnett squares. For the last part of this chapter, we will cover non-Mendelian genetics. These are inheritance patterns that are more complex than those predicted by simple Mendelian genetics. The relationship between genotype and phenotype is rarely as simple as in the pea plant characters Mendel studied. Many heritable characters are not determined by only one gene with two alleles. However, the basic principles of segregation and independent assortment that he discovered do still apply to more complex patterns of inheritance. Inheritance of characters by a single gene may deviate from simple Mendelian patterns in the following situations: when alleles are not completely dominant or recessive, when a gene has more than two alleles, or when a gene produces multiple phenotypes. Let's look at all of these situations. There are varying degrees in dominance. The type we've discussed in Mendelian genetics is called complete dominance, when the phenotypes of the heterozygote and dominant homozygote are identical. We have two additional types of dominance, incomplete dominance and codominance. We see an example of incomplete dominance shown here with the red, white, and pink flowers. The phenotype of F1 hybrids is somewhere between the phenotypes of the two parental varieties. Look at the flowers in the P generation. The red flower has two identical alleles that are CR, while the white flower has two identical alleles that are CW. Notice that there are a few ways that alleles are depicted in genetics. This is a capital letter with a capital superscript. The F1 generation received one CW allele and one CR allele and exhibits a pink phenotype. When you cross the F1 generation by itself, as shown in the Punnett square, look what happens. The red and white flowers reemerge. We also have two pink hybrids present. An example of codominance in humans is hair. Curly hair crossed with straight hair gives wavy hair, the heterozygote. In codominance, two dominant alleles affect the phenotype in separate distinguishable ways. Your blood type is governed by codominant genetics. The three alleles for the ABO blood groups and their carbohydrates are A, B, and none, also called O. The letter used to denote these alleles is I. Both A and B are dominant while O is recessive. An A allele is a capital I with an A superscript, while a B allele is a capital I with a B superscript. The recessive allele is a lowercase I. A person that has a phenotype A can have two different genotypes, either big I A, big I A, or big I A, little I. The same is true of the B blood type, big I B, or big I B, little I. A blood type of AB contains both dominant alleles, so this genotype is big IA, big IB. Remember, this is codominance. Lastly, an O phenotype individual has a genotype of little I, both recessive alleles. While we've been discussing genes very simplistically, in a way that makes it seem like one gene controls one character, most genes actually have multiple phenotypic effects. This property is called pleiotropy. Pleiotropic alleles are responsible for the multiple symptoms of certain hereditary diseases such as cystic fibrosis and sickle cell disease. And some traits are determined by more than one gene. Two examples shown here are epistasis and polygenic inheritance. In epistasis, a gene at one locus alters the phenotypic expression of a gene at a second locus. In this Labrador example, 3 coat colors are possible: black, chocolate, or yellow. There are 2 genes that determine color, B and E. If the dog has a dominant capital B allele, it will be black, while if it is homozygous recessive for B, it will be brown or chocolate. However, no matter what the B alleles are, if the dog is homozygous recessive for E with 2 little e's, the dog will always be yellow. In polygenic inheritance, quantitative characters are those that vary in the population along a continuum. There's an additive effect with these genes. In this example, we see skin color. The more alleles for darker skin color, the darker the skin shade. Height and body weight are additional examples of polygenic inheritance. We can't discount the impact environmental factors have on our phenotype. Another departure from Mendelian genetics arises when the phenotype for a character depends on environment as well as genotype. This is the classic nature versus nurture debate. The norm of reaction is the phenotypic range of a genotype influenced by the environment. Norms of reaction are generally broadest for polygenic characters. Such characters are called multifactorial because genetic and environmental factors collectively influence the phenotype. Examples of multifactorial characters are body weight, blood pressure, and blood sugar levels. The last thing we will cover in this chapter is pedigree analysis. A pedigree is a family tree that describes the interrelationships of parents and children across generations. Inheritance patterns of particular traits can be traced and described using pedigrees. Take a look at the legend across the top. We see females are depicted as circles and males as squares. Shaded in shapes indicate that that individual has the trait that we are following. In pedigree A, we see that the male individual in the first generation who has the trait has a heterozygous genotype of big W, little w, while the female in the first generation who does not have the trait has a homozygous recessive genotype of little w, little w. Look at the second generation. What are the genotypes of the individuals with the trait? Can you tell by this pedigree whether this trait is dominant or recessive? This trait, widow's peak, is dominant. Quickly, notice the individual in the third generation with widow's peak. Can you tell what the genotype for this individual is? We know that they have the trait, which means that they carry a dominant big W allele. However, there's not enough information to figure out whether this female would be homozygous dominant or heterozygous. Can you figure out a way that we'd be able to tell? Now take a look at pedigree B. Can you tell if this trait, the inability to taste PTC, is dominant or recessive? All individuals with the trait have the genotype t, homozygous recessive. Therefore, we can say this trait is a recessive trait. Note that both of these traits are called autosomal traits. They are carried on genes on autosomal chromosomes. Remember, autosomal chromosomes are all chromosomes other than X or Y. X and Y are the sex chromosomes, and we will see more about sex-linked traits in the next chapter. We will also see more types of pedigrees. Pedigree A is an autosomal dominant trait, while pedigree B is an autosomal recessive trait. Take some time to attempt the practice questions prompted throughout the lecture. It's very important to understand the concepts in this chapter before moving on to the next chapter, which is the chromosomal basis of inheritance.